All right, well, thank you to the sponsors for letting Luminary speak, but more importantly, thank you for the people that are here to listen. I know it's getting close to the end of the day, and I'm sure cocktails are starting to get on everybody's mind. So Luminary Therapeutics is all about unlocking access to CAR T therapies. So I think it's the ARMS data that suggests that only 20% of patients that are available to get CAR T therapies get them. And we all know the reasons why. Most of our current CAR T therapies are autologous, and they're costly, they're made in centralized manufacturing centers, and they're hard to get to patients, and unless you're near a large cancer treatment, you're not going to be able to get these treatments. So our vision is to make them affordable and accessible. So when we started this company, which we had a previous company that we built a gene editing tool, we wanted to focus on some of the bigger challenges. The supply chain, of course. We went into this company 5 years ago thinking about autoimmune diseases before it became the most popular rage. And then, quite frankly, we want to be one of those companies that is going to knock the door down for solid tumors. And when I get to that part of the presentation, I'll tell you why we think we can make that happen. I'll go back for just a second. So when I talk about making them affordable and accessible, we're not bashful about saying that effectively our allogeneic products we can make for under $10,000. And we think we can move the industry forward in dropping the cost and the price of these cell therapies from the $450,000 down to $185,000. So how do we do this and how do we go about this? Well, we have an allogeneic manufacturing platform where we use gamma delta cells, and I'll speak about that a little bit more, and we're able to get 450 doses out of a single healthy donor. We can take those 450 doses, manufacture them into just 2 clean rooms, and run them on 2 shifts, and we can make 50,000 products a year, which is more than patients have been treated to date in the entire CAR T industry. For our autoimmune program, one of the things that we pride ourselves on is we bring out new novel targets, and we never bring out a single antigen target. So all of our targets are multi-targeted. I'll talk about, again, how we're approaching solid tumors when I get to those slides. So I'm backed by a bunch of really strong, talented people. The scientific founders of our company are Brandon Moriarty and Bo Weber. We had a company together, so the 3 of us have been working together for 9 years. They're University of Minnesota professors and world-renowned gene editing expertise. We partner with a Dr. Paolo Camille out of the Cleveland Clinic, who's just been tremendous in the 5 patients that we've treated to date. Our chief medical officer is fractional, but his name is Matthew Spear. And then we have a bunch of regulatory and quality people. And then our head of operations is a lady named Linnea Mead. So, as I noted, we are a clinical stage company. These are the inclusion and exclusion criteria of the trials that we're currently running. So we're in non-Hodgkin lymphoma, we're in chronic lymphatic leukemia, and we're in multiple myeloma and lupus. This is all with the same product, so this is, even though we're running multiple trials, it's just a single product. As noted, we've treated 5 patients to date. We have data on the first 4 and are starting to get data on the 5th patient. So I'm happy to say that we've had a complete response. We've had an overall response of a very high rate. We've treated some very tough patients that are 4 and 5 lines of previous treatment. So we should get some data on our 4th patient probably in the next 3 weeks, but we're hopeful that we'll have a complete response there. Our lupus patient is effectively scheduled to manufacture the product on May 13th of this year, so we will join the groups of companies that have treated lupus with CAR T therapies. So what are our cell manufacturing advantages and how do we do this? How are we able to get to a $10,000 cost of goods sold? Well, the first and foremost is we use these gamma delta cells, and unlike the other gamma delta cell therapy companies out there, we use both subsets of the V delta 2 and the V delta 1, and this is an extremely powerful combination If you look at the way, we were built in nature, we don't separate those two particular immune cells, so we decided our final product should consist of both of those. V delta 2s are very much like NK cells, rapid shock and awe response, don't stick around, but V delta 1s will stick around and will hone to the tissue. We also cloak our cells to make them hypoimmune. We got this technology out of Mass General Harvard from Marcella Moss. So we're able to put in cells into a recipient's body. They're not viewed to be foreign, and yet they are still highly functional. And then additionally, we use our non-viral gene editing tool that we created at our previous company. So this allows us to get clinical grade GMP reagents in 4 months. And we can put in larger cargo, safer insertion rate, and effectively get 50 to 60% integration. So in addition to that, part of our proprietary technology is once we've engineered this dual set of gamma delta cells, we're able to expand those 3,600-fold. And we have some very special technology to do that, as well as a lot of know-how of what cytokine cocktail to put in, when to put it in, how to use the G-Rex product to effectively let those cells grow and expand. So as noted here, some of the data on various manufacturing runs, various CAR constructs, where we've been able to get this 450 patient doses. And that's assuming a 350 million CAR positive cells for the dose. So our autoimmune franchise, obviously it's a bigger market that everybody is pursuing in the CAR T therapy, so we're not the first to go there, but we think we have a better product, and I'll explain why. So we know that the challenges of CAR T therapies in autoimmune is the fact that long-lived plasma cells, which are the cells that kick out the preponderance of these autoantibodies in creating this inflammation, et cetera, are there and exist. And that little graph that might be a little bit hard to see shows patient data and effectively the quantity of long-lived plasma cells. The other thing, especially for lupus patients who have been on high regimens for rituximab, over that, they don't have very many T cells to work with. So if you're an autologous manufacturer, you're going to have trouble making that product. And then the demand that this will place, we think that there's, for the autologous companies, going to be a challenge there. So here's our secret sauce. We have a CAR, and as I mentioned, we only bring novel targets to the clinic, as well as we don't bring single antigen targets. So on the left-hand side, you can see a normal CD19 CAR T cell with a single receptor. Our CAR is ligand-based, and we're able to load that up with 3 receptors. So we have BAFF, TACI, and BCMA. And BCMA is what is expressed on long-lived plasma cells, and they stop expressing CD19. So our belief is we will have a complete reset of the B-cell compartment all the way into the bone marrow and the tissue, and therefore we'll have a much more durable response than the CD19 people. As noted, I talked about rituximab, so here's a rituximab patient sample of the T cells available to go ahead and make a CAR T product, which is very limited. We've already tested this platform on our allogeneic program and effectively showed clearance of all B cells from lupus to systemic scleroderma to dermatomyositis. We know that this product is safe. As I noted, we've treated 5 patients to date. We've only had 1 level 1 CRS, and in our second patient, we had a complete response in our oncology trials. So we feel that the data right now is promising. We're looking to get 10 to 12 more patients treated in 2025 to see if these statistics hold, and if they do, we think we're well on our way to a new novel product that will challenge the CD19 world. So now when it comes to solid tumors, how do we approach this? And we know that this is a tough nut to crack. So we have 3 different ways that we're trying to attack this particular angle that can work in unison or work independently. So the one that we have presented the most in the past is this dual CAR T concept. A lot of people have different approaches with using dual CARs. Our particular dual CAR, which I'll show in a slide, has some very unique signaling properties, that give it optimal fitness and durability. The other thing that we were just awarded an ARPA-H grant for $6 million is gamma delta cells actually can act as professional antigen presenting cells. So we have a program where we're mixing a CAR with many tandem genes inside of our construct, inside of our transgene, and we're able to load that up with 50 to 60 neoantigens and use the gamma delta cell's ability to effectively present that to the endogenous immune system and go after that. And then finally, we have a program where we have a CAR construct that has a couple of receptors on it that do the following. They can break up the tumor microenvironment by eliminating those fibroblast cells and eliminating those myeloid suppressive cells. So we believe strongly that one of these three things, if not multiple ones, are going to kick down the door, and CAR T therapies are going to finally have their day in the sunshine for solid tumors. So it's a bigger market, it's less crowded than the hematological space. As noted, here is kind of all the challenges of a tumor microenvironment. I'm sure most people in this room are highly familiar. But effectively, this dual CAR that I mentioned that we use, it does a couple of very unique things. Is we share the activation domain for both of those CARs, and then we split off the co-stimulatory domain. And I don't know if this thing has a pointer. It looks like it might. But if you see this little yellow bar, so we get this dimerization impact that happens in the transmembrane, and that gives these CAR cells an extensive amount of durability and make them optimally fit. So here's kind of the proof in the pudding. We've taken multiple types of dual CARs, B7H3 with GD2, CSPG4 with meso, and shown and demonstrated that in mice we can get clearance all the way of a variety of solid tumors 100 days out. Again, noted that this ARPA-H award we have, where we're able to take a CAR, so we get the CAR part, and then we load up that CAR with a mini tandem gene that has a bunch of neoantigens. So here we're pursuing the whole essence of the heterogeneity of a tumor microenvironment. And we're very excited about this. We're just have engineered these cells in the last couple of weeks, and we'll start to take those into both in vitro and in vivo. And the best thing for Luminary is this is funded all the way through IND with non-dilutive funding. I don't have a whole lot of cells on our OSM CAR that knocks out these myeloid and suppressive cells because that's pretty new to us. We always try and check ourselves at the door to make sure we're not drinking our own Kool-Aid, but we've been fortunate enough to be recognized both by AbbVie, Bayer, J&J, NIH, and we feel like we've accomplished an awful lot since the inception of the company in 2019 to today. As noted, we've treated 5 patients in the clinical trials. We have 3 phase 1 trials going on, and we've done this on a very modest budget, I can assure you. So that's us in a nutshell, and again, thank you for sticking around.